different, and there's slightly different makeup to them here. But we're going to use terms, or at least prefixes for it, that we have used before. Endo and epi, and we'll use myo in the middle, but looking at the order in which we cover them here, here is the inside of a ventricle, right here, and the inner lining of the heart is called the endocardium. Now, the endocardium, we'll look at it in just a little bit here in terms of more detail. But then the middll layer, the muscle layer to speak of, which will contain the Purkinje fibers in it as well, is called the myocardium, myo meaning muscle, of course. Then the outer layer of it, we'll use the prefix epi, just like we've used before. The epicardium is the outside here. Now, let's take a look at each of these in a little bit more detail on the next couple of slides here, starting with the endocardium. Here is the endocardium, in this case the hollow of either the ventricle or the atrium. Actually, it's going to be a ventricle if they're saying these are Purkinje fibers. The hollow of the ventricle is right up here. And so here is that inner lining. If you take a look at it, it's fairly high power magnification, 300. It's got a simple squamous epithelium lining. Kind of miss it until you just look at those pancake nucleus, one row of them there. And then a very thin layer of underlying connective tissue before we get into the pink myocardium down below right here. That myocardium is what is composed of cardiac muscle, and they don't really show this that well to say, oh, those are indeed Purkinje fibers. That is, in fact, muscle. They were interested in looking up here at the endocardium, and thus didn't choose a slide that showed this very well, but this shows the myocardium like you are used to seeing it. Remember, when you see cardiac muscle in cross-section, there are islands, and when we see the nuclei within those pink islands, those nuclei are going to be more centrally located, not on the coast, and they don't take up a huge portion. They aren't the majority of that island like it was in smooth muscle. In terms of longitudinal section, like we see on the left right here, you see the intercalated disc staining a little bit lighter there. You see that woven type of appearance, and you get a little bit of a hint if you look in certain places in here, like where the pointer is. Right in here, I get the impression here of striations. I can see them a few other places in the slide there as well. Now, the myocardium, like we said, is the muscle layer. It's going to be the thickest of the layers. That's the, I'll say, functional layer of the heart. That heart beats due to the muscle that is there. There is going to be a difference in the thickness of the myocardium depending on where we are within the heart. If you think back to anatomy last semester, atria, they don't have to pump blood that far just into the ventricles, right? They just make certain the ventricles are filled before they pump, whereas the ventricles have to pump either through the entire pulmonary circulation or through the entire systemic circulation, in which case the ventricles have to have a much thicker muscular wall. And of the two, which do you think has the most work, pumping through the pulmonary circuit or through the systemic circuit? It would make sense, systemic, and so of the two ventricles, left ventricle is going to have the thickest wall, correct? I mean, just common sense if you think through it like that. Last thing I've got in the notes, we talked about cardiac muscle, rich capillary supply. They've labeled a few of them in here, but any of the bright pink you see in here will in fact be capillaries. Now they've got these two labeled here and here, but here, 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 I mean all over, here. All of those are capillaries, you already know. Very rich capillary supply there. The outermost layer will be the epicardium, and the epicardium is going to have can't see it that well on here because we're at too low of a power magnification, but there will be a simple squamous epithelium on the outside. We often refer to it as a mesothelium on the outside there. And then depending on age, also depending on where we are within the heart, there may be large amounts of adipose, like you see this slide on the left. If we were to look at this in a newborn, not only would it be adipose, it would be brown fat instead of unilocular adipose, or it may just be fibrous tissue right here. Now this epicardium is synonymous with the visceral pericardium that we talked about in anatomy. Okay, if you remember you had a parietal and a visceral pericardium. It reflected back on itself. They were continuous, and that visceral pericardium was on the outside of the heart. That is the same thing as the epicardium. Okay, gross anatomists call it the visceral pericardium. Histologists call it the epicardium. All right, let's talk about a few other features that we can see. First slide we looked at here, we can see some papillary muscles inside the ventricle. That's how I knew this was a ventricle before, is the papillary muscles. And you know the papillary muscles will provide tension to the chorda tendineae, which in turn prevents prolapsing of your atrioventricular valves. Occasionally you can see chorda tendineae coming off of the papillary muscles. It just looks like threads coming off there, a little piece of cord, let's say, coming off there. Very seldom you actually catch it, but every now and then you do. More likely just to catch a piece of it somewhere in there, but every now and then I've seen a few slides in textbooks where you have a piece of it coming off the tip of, say, the papillary muscle that's labeled there with P. The valves are something else that we obviously can see histologically, and this is looking at valves. We look at the larger image first of all. Here's a valve coming off from the left atrium, left ventral. Here's the valve coming off the edge of the wall. They don't show it all the way out, but you do see the, I'll say the skeleton of the valve, the structural component that keeps the valve intact is called the tunica fibrosa. It's that pink layer in there. It's got a lot of collagen fibers in there. Collagen fibers, you think of that as like leather when they're woven properly, and that would indeed make good sense. This is a leather-like structure, so it's got some flexibility and yet some strength to it. Now when we stain it with an elastic stain like we've done on the two images on the left here, you can see the fact that there's elastic fibers up here, a good bit of them that are kind of staining the greenish color. You got some down here, but you have a great deal more up here on the atrial side of that valve. And here we just took what this bottom image did, was just a higher power image of this portion up here to show those elastic fibers. When you think about it, that valve will bend into the ventricle, okay, and then come close back when that ventricle contracts. These fibers are up here on this surface right here, so that as that bends, it stretches those elastic fibers, and those elastic fibers then help close that valve along with the ventricular pressure that's created when that ventricle contracts. All right, and should point out, I've got it written there in the notes, realize if this is the atrium, it's lined by an endothelial lining, and simple squamous endocardium lining up here. It's going to continue on the valve. The same thing with the ventricle. There's endocardial lining right here. And so that valve is lined on both sides. The surface on both sides has an endocardium lining it because that valve's continuous with the wall. All right. Now, in terms of the cardiac conduction system, there is not a whole lot that you see with this. We can see the atrioventricular valve and the node and the sinoatrial node. This textbook that we use in this class, I want to say about 4 or 5 editions back, they actually show the SA node and AB node. I don't know why they took it out, but that's the only textbook I've seen it in. I mean, I went and looked online for images of it. The only images I could find were from journal articles. I said, okay, don't really need to show it. It does look different. When we looked at cardiac muscle, it had kind of that woven appearance to it with those Y-shaped cells. There's a lot more collagen fibers at these nodes, and rather than the woven effect, the myocytes are more swirly, okay? A swirling effect to it versus that woven effect is what you see it, and there's not the open space that looks in between there because it's filled more with collagen fibers. And so you can't necessarily correlate, that's why it's the SA node or AB node. It's just we know that when you look at the nodes underneath the microscope in section, that's what they look like. I mean, I can't give you a structure-function relationship there, in terms of why it is the way it is, why it's got these collagen fibers, why it's a swirling effect, and so on. What we can see though is once we get past that SA node, that AV node right here, once we get here to the bundle of his right and left bundle branches, and then all the way down here, that is going to be composed of Purkinje fibers. Now this can sometimes be confusing to students because, on gross anatomy books like this, we label typically the end branches down here as being Purkinje fibers, but it turns out everything from here on down is composed of Purkinje fibers. They're just larger bundles of Purkinje fibers. Whereas these, when you get to the terminal ends, look like just linear Purkinje fibers lined up. If we get up here within that interventricular septum, the components there look like big bundles of Purkinje fibers, and this is often where we look at them histologically. Simply they're larger, easier to find. You know exactly where they're going to be. They're going to be right deep to that endocardium, okay, right within that interventricular septum right there. Now, you know what Purkinje fibers look like. We've looked at them before. Once again, this would be the hollow of the ventricle up here off the top of the screen. Here's my endocardium. With a little enough power, I can't make out my endothelial, my simple squamous epithelial lining up here. And then right here, just deep to it, that is where we're going to find the Purkinje fibers. And no, I don't expect you to look at this and say, oh, those Purkinje fibers. If we were doing this on a microscope in lab, I'd have you find that septum, look just deep to the endocardium, and then right there is going to be one of the bundle branches, and go to higher power on that, and here's where we're going to see the slide we've looked at before for the Purkinje fibers. Doesn't always show great striations in there. I showed you one in lab that did, but the things you recognize it by position, first of all, just deep to that endocardium, and then also the fact that the nuclei have perinuclear glycogen stores, so they often have a negative staining image here, a washed out image surrounding the nucleus. Okay? All right, that takes care of the heart. What we want to do now is move into blood vessels. Okay, whereas in the heart we had cardium and prefixes in front of that, endocardium, myocardium, epicardium, to name the layers. We're going to name the layers differently within blood vessels. The word we're going to use is the word tunica, okay, and if, Old English, so you're talking about, Shakespeare or prior to Shakespeare, a King James Bible, what's a tunic? It's a coat. Okay, and so we're saying here there's different coats. There's a tunica intima, a tunica media, and a tunica externa. Those are the 3 layers that we can find within blood vessels. Tunica intima is an intimate contact with the blood, and so it is the inner layer right there. It's composed of endothelial cells, which are simple squamous epithelium. We just give it a special name when it's lining blood vessels or lymphatic vessels. Rather than calling it simple squamous epithelium, we refer to them as endothelial cells. I use that term, didn't mean to, but I use that describing the simple squamous epithelium of our endocardium as well, and that's a legitimate use of it as well because it's continuous with the blood vessels. All right, then there's a thin layer of underlying connective tissue. We'll see that in more detail underneath the microscope here in subsequent slides. Then the middle layer, which will contain smooth muscle, is tunica media, media meaning middle. And you can also think for that M in media as standing for muscle. It's going to be smooth muscle, of course, not cardiac muscle like we have within the heart. And it's going to form, the cells are going to be oriented in a ring-type manner surrounding the lumen, circumferential around that lumen, such that when they contract, they're going to make that lumen constrict. All right? And then the outer layer is called the tunica adventitia. Another term that is creeping in is calling the tunica externa. And this can differ depending on where we're talking about. You know, right here, they're showing a large artery on the left, a large vein at the right, large enough that you can dissect it out and hold it between your fingers, in which case you're going to find just a little layer of connective tissue and then a simple squamous epithelium on the outside. But if we're talking about a smaller blood vessel, you know, running in your biceps, it's going to be running, let's say, through paramecium of your biceps. And in that case, that outer layer, that tunica adventitia, really is not a distinct layer, it just blends into that connective tissue it's running through. Okay, and that's what we mean by an adventitia. Okay, we get to other organs we'll talk about. Sometimes it's got a serosa, a defined outer covering. Other times it does not, it just kind of blends into surrounding connective tissue. That's an adventitia. And so, you know, there are, I've seen even a few sources out there that will talk about it being a tunica adventitia, you know, when it's small, running through the paramecium of the biceps, and a tunica externa when it's, you know, large enough that you dissect it out and hold it between your fingers. Okay, and so synonymous terms used a little bit differently sometimes, but it's not complicated, it's not tricky in terms of understanding that and understanding why you've got different terms in there, why one source chooses one versus the other. All right, if we take a look at it in the drawing here on the right, different source here, but if we take a look at it, we can see here's our tunica intima, and we can see it's got multiple layers to it that we'll define here in just a little bit. Here's our tunica media, smooth muscle, the fibers oriented in a ring around the lumen. And then out here is my tunica adventitia, which has connective tissue, and if it's not running through connective tissue, if it's something you can dissect out as a separate organ in and of itself, it would have a simple squamous epithelium on the outside here. Now this is labeling some of our extra components, internal, external elastic membrane, and so on. We'll look at those in more detail here as we go through, particularly within the arteries here. Arteries, you know, are blood vessels that carry blood away from the heart. That's our definition of arteries. And because they are carrying blood away from the heart, they're under the most pressure, and so they're going to have the thickest walls in comparison to veins. And we'll certainly emphasize that when we look at arteries and veins right next to each other. All right, now the largest of arteries are what we call the elastic arteries. They've got elastic fibers woven into sheets, and it makes it so that when the heart pumps blood, I mean, the heart doesn't pump blood like a water faucet when you turn it on, how it just has a normal stream. I think we've all seen gory movies, whether we wanted to or not, where someone was shot or stabbed in an artery, and it pulsates, doesn't it? Okay, and that's because when that heart squeezes, you're going to have blood squirting out, and then it's going to have to refill, and the heart, you know, basically pumps in spurts like that. Well, if it's pumping through that aorta in spurts, what the aorta does, because it's an elastic artery, is the fact that when the ventricle squeeze, the aorta comes back out, and what's going to happen then with these elastic fibers on the wall, when the heart is refilling, it's going to recoil back in, won't it? Kind of helps make it where I don't reach a high spike blood pressure as I would normally, but also my blood pressure doesn't go as low as it would as my heart is refilling. Okay, and so it kind of helps moderate blood pressure here with the elastic fibers here within the walls of the elastic arteries. And whenever they show elastic arteries, they're always going to show the aorta. I mean, we could go a little bit, we could do carotids and things like that, but aorta is the one they're always going to show. We look at it here, very low power magnification in the image on the left. You know, it's magnified 33 times, you can see below, so I cannot make out my endothelial lining up here. They're labeling this tunica intima. Where they happen to draw that line right here, it was just, that's where I decided to draw it. I mean, somebody may have drawn it, you know, a little bit over here, somebody may have drawn it a little bit further this way, but I could see a lot of people wanting to draw it maybe right in here instead. And so don't get caught up, how can I tell the difference between there? And you won't have to define exactly where what's the border here between intima and media, okay? But if we take a look, this is stained with an elastic stain. And you see the elastic fibers are primarily here within tunica media. I do have some out here in the tunica adventitia. And when they go to a higher power magnification, almost 10 times what we had in the image on the left, you can see these large diameter collagen fibers here. Once again, they're woven almost as a sheet of fabric. Yeah, synonymous terms. Tunica adventitia and tunica externa will be synonymous terms. Okay, technically would be a difference, but in old histology textbooks it was all tunica adventitia, even when it truly wasn't an adventitia. Okay, now the elastic arteries will contain also extra elastic fibers within tunica intima, and what we're labeled back here as the internal and external, or should I say the internal elastic membrane. They show up, they're here, but there's so many elastic fibers present. They show up much better in a muscular artery, which is the next one here. And here is what we're looking at right here. Fairly low power magnification, 100 times. Even with H and E, the elastic fibers here, if you look at, there's a endothelial, simple squamous epithelium lining, a little bit of connective tissue, and just deep to that, there is, if we take a look at this pink line running right here, if we look with an elastic stain, this dark line running right here, that is what we call our internal elastic lamina, or it can be called an internal elastic membrane. And I tend to use those terms interchangeably without thinking about it. All right, and that's what they're pointing to with the arrows. Now, how do you know that what that arrow is pointing to? I thought it was pointing to tunica intima or my endothelial cells. I won't give you both as a choice. I can only give you one of those choices on an image like this. Now, so there's this right just underneath that endothelium is our internal elastic lamina. There's also an external elastic lamina right here. And I think it shows up better over here in H and E. You can see this squiggly line right here, where those elastic fibers will pick up H and E if it's the right formula of H and E. That is the border between the tunica intima and the tunica media. Some books will list it as the border between. Some books will say it is the limits of tunica intima. In other words, it's in tunica intima. Others will say it's the border between intima and media. Don't get caught up in that. Doesn't really matter. Both are describing where it is, correct? It is the border right there between the tunica intima and tunica media. And yes, it is there within the elastic arteries, but there's so many elastic fibers in there, we have trouble seeing it, okay? If we went to higher power in this image right here, we would at least be able to make out that that internal elastic lamina. External, there's just so many within the media already that we'd have to say it's just the first one we see within that media. The other side, it is right here. If you look, this little squiggly line right here is my external elastic lamina. That's the border between media above and our intima above and media below. Oh, whoa, I've got something wrong. Thank you. Let me go back to the drawing. Thank you. I'm going to have to correct myself. My internal elastic lamina is a border between my tunica intima and my tunica media. Okay? That's where it is located. My external elastic lamina is out here. It's the border between media and adventitia. My complete error, what I was saying previously on that. And thank you for catching it. I should have caught it because there's a nice smooth muscle layer right there in there, okay, of tunica media. And so there's my tunica media, here's my tunica adventitia out here. So internal elastic lamina, border between intima and media. External elastic lamina, border between media and adventitia. Okay? I think I typed that in correctly and spell check didn't catch me, okay? That should be external, not eternal, all right? There is a difference. Yeah. Haven't caught that because spell check didn't underline that for me. All right, now let's talk about muscular arteries, and I'll leave it on this because this is a muscular artery right here. Muscular arteries are also known as distributing arteries, and they're both very good names for them. Muscular arteries, they're described because the tunica media is very prominent, and it's not so full of elastic fibers that it, covers up the muscle there, hides that muscle. The muscle's a lot more evident because it's just smooth muscle. There's very few elastic fibers there. Distributing arteries because the branches off the aorta, the superior inferior mesenteric, the splenic artery, the hepatic artery, the renal arteries, all those are muscular arteries, and they distribute the blood. What do I mean by that? Well, I just ate a breakfast burrito right before coming in here. My inferior and superior mesenteric arteries are going to be dilated to send more blood to my intestines to digest them, correct? On the other hand, if I was right now running on a treadmill, I need extra blood going to my lower extremities here. I'm not going to send so much to my internal organs, my interior, superior and inferior mesenteric arteries, my splenic arteries, and so on are going to be constricted, sending less blood there, so more can go to my legs. Now, if I just ate that burrito and then I jumped on the exercise bicycle, why am I sick? I don't have enough blood to go both places, correct? I'm sending all that blood to my legs, and my GI tract is not happy because it's working and not getting enough blood to work. Okay, does that make sense? That's why they're called distributing arteries, because they can control where you distribute that blood within the body. Okay, and so like I say, both of them very descriptive. Questions?
So at the top of those pictures, is the tunica intima just the white space?
We're at such a low power magnification on both of these that we're not seeing tunica intima.
Okay.
I mean, tunica intima is on the above that internal elastic lamina. Okay? And that's why I say I won't give you both as a choice if I was labeling that with the arrow and say name what's labeled by the arrow. You can't tell me for certain what it's pointing to. All right, now the muscular arteries are the ones we often will use when we're going to show the various tunics, simply because they show it the best. Even smaller muscular arteries such as this right here. This is still considered a muscular artery. Once again, that tunica media is very pronounced in it right there. You can see the idea that it's running through connective tissue on the outside, but my tunica intima, it's just going to be on the surface. You can see some of the nuclei of those endothelial cells on the surface right here. And they're labeling the H&E here that's been modified a little bit to stain the elastic fibers. I can see my internal elastic lamina a little bit. And then my tunica media, very prominent right through here. And I've got a little extra pink on the outside. That would be my external elastic lamina, although it really doesn't show up that well. But then outside of that, I'm into my tunica adventitia, which will just blend into the surrounding connective tissue. You can see it a little bit better over here. It's about double the magnification power, a little over double the magnification power of it. But here's my lumen. You can see the E is labeling those endothelial cells of the tunica intima. And then all of this is smooth muscle cut in cross-section coming out of the screen at you. Same thing up here. And then out here is my tunica adventitia. Okay? And so this is, we'll look at some more slides like this in lab. This is a typical small muscular artery that we are seeing right there. Now the muscular arteries are eventually going to branch into smaller arteries which will eventually go into arterioles. Arterioles are the most distal branches of the arterial system, and we talk about there's still smooth muscle left there. There's still a tunica media. When there's no longer tunica media, will be in capillaries. These are going to be very important in terms of controlling blood pressure. I believe I talked about that when I talked about these in anatomy. If you walked in and didn't realize there was a test this morning and your blood pressure spiked up, that's because your arterioles went like that, okay? And of course, we often will use medications against them to keep them dilated to lower blood pressure. Also very important in controlling blood flow, and we'll talk about that when we talk about medarterioles here in just a minute. Before we do that, let's look here at the idea of an arteriole. We've got two arterioles right here. You can see they're running through connective tissue. But if you take a look at them, you can see the endothelial cell lining in certain places here. And then if we look in the wall here, it's about, look right here, maybe 1, 2, 3 layers of smooth muscle cells thick. It could be one for all that matter, and it could still be an arteriole, but it's just a few cell layers thick. It's not the 8 or 10, whatever in the previous small muscular arteriole we talk about. Here's an electron micrograph showing them. Now when you look at this, here's the endothelial cell nuclei. Normally simple squamous, we talk about the nuclei being flattened out like pancakes. What happens though is when we're making the slide, basically it constricts, and as it constricts, it then bunches up those endothelial cells, so they're not a true, they don't have the normal shape of a simple squamous, and that's why you often will see, like the nucleus right below my pointer here, that's an endothelial cell nucleus, but it certainly is not pancake with long axis parallel to the surface anymore, is it? And that's got, you'll see that a lot of times in blood vessels, but you know by the location that these are my endothelial cells, this is my tunica intima, and basically these are the nuclei of smooth muscle fibers here in the wall. If we take a look, this is only one layer of smooth muscle fibers thick in terms of its tunica media, isn't it? Okay, whereas this one is maybe 2, this one may be 3 in some places here. Now the smallest of arterioles is what we call a metarteriole. Here's the arteriole in terms of the drawing. If you take a look, that smooth muscle is continuous. They're not really showing the tunica adventitia on the end, they're stopping at tunica media. But then it branches off here, and this is the metarteriole. The metarteriole is just before it goes into the capillaries. And the smooth muscle may be discontinuous. It's not necessarily, if you look here, the smooth muscle fibers are not continuous like they are up here. Okay? The metarteriole is important because it controls the blood flow within a capillary bed. If you pull up a video, you can easily Google YouTube video, blood flow capillary bed. What you're going to see is it's not a continuous blood flow. Okay? If you look and you have the microscope on the capillary bed, how would you do this? It's not easy, but it can be done in a freshman biology lab. A lot of people try to take a frog and spread its toes apart and put the webbing on the slide, and then you can focus and actually get on the capillary and see blood flow. There's an easy way of doing it. If that frog is moving around too much, a minnow, use a fin on it. And if that minnow gets out of the water longer, he doesn't move as much, you can still put him back in the water and he's going to be fine. You obviously don't leave him on that slide too long. But if you look there and you've got on the capillary, you're going to see blood. Instead of blood, flowing along and flowing along the capillary, it'll stop and it may kind of move a little bit. And all of a sudden it'll start flowing again. And basically what you've got here is the cells here, in between this network, meshwork of the capillaries. Basically, they're going to say, I need oxygen. That metarteriole is going to dilate. And when that happens, you're going to see blood flow going through. Then those cells are going to say, I'm fine, I've got enough oxygen. That metarteriole will constrict and not send as much blood flow through there. And that's when the blood doesn't move, or maybe just ebbs a little bit forward or ebbs a little bit backward. The cells, wait a minute, need more oxygen again. And that precapillary sphincter there, that metarteriole will open up again. And it's doing that every 20 seconds or so in a resting state. Realize if you're on that exercise bicycle, those capillaries in your muscles, those precapillary sphincters, those metarterioles are going to be completely dilated, correct? It just makes it so that we can control based on oxygen needs, as well as we can say other metabolic needs, buildup of waste products, buildup of acidity, things like that. Various metabolic factors will feed in to that metarteriole to determine does it need to dilate or does it need to constrict based upon needs of the tissues downstream.
Are these cells releasing chemical stimulus to tell the metarteriole cells to close, or?
I would leave that to the physiologist, okay? It can be local metabolites and it can be oxygen levels, okay, and I can't tell you what all those metabolites is. pH would be a very good one. But I don't know exactly, and my physiology is very outdated compared to Dr. Oldie. I just don't want to tell you wrong, and then you have it wrong with physiologists. All right. The next thing to talk about then will be the capillaries. And capillaries are defined as blood vessels that consist only of a tunica media. I'm sorry, a tunica intima, okay? They have no media to them. This is a capillary. This right here is a capillary running across the screen. You can see the red blood cells in it. And we look at the wall, basically all I've got is an endothelial lining. If I say, wait a minute, it's underlying connective tissue, well, that just blends into the connective tissue it's running through, okay? And so all it has is an endothelial lining. Here's a capillary coming out of the screen at you. There's a red blood cell right there in the middle of that capillary. And you can see this is one of the smallest capillaries. It's just one endothelial cell that its arms reach out and touch each other. Other capillaries would have, endothelial cell reaches only out this much, and I've got two other ones making up the rest of the wall for larger capillaries. But capillaries only have endothelial cells making up the lining. And of course, capillaries is where you have all chemical exchange between blood and tissues. And I say it goes both ways, tissues to blood. Oxygen is going to diffuse across the wall of the capillary. That's why you have a nice, thin, simple squamous, really thin, simple squamous epithelium endothelial cell making up that wall. And that's where CO2 will diffuse back into the capillaries from the tissues. Now, there's also another cell. If we take a look, here's my endothelial cell. I've got another nucleus right here. Same thing down here where they've labeled it with a P, another nucleus just outside. A little tiny bit of pink cytoplasm. I want to say, oh, is there just occasional smooth muscle fibers in there? No, that's not the case. This is another cell that we call a pericyte, not a parasite, but a pericyte here. And these play a similar role to smooth muscle. They play a contractile function, but you look, they're kind of scattered throughout. It used to be, 10 years ago when we were talking about these, we thought maybe they were a stem cell that could provide replacement. Turns out that's not the case, okay? You may still see that they're there for repair or stem cell of some kind. That's just a histology website or histology textbook that hasn't caught up with the research that came out about 10 years ago. Okay? Now, it turns out there are different types of capillaries. The capillaries that you think about typically are what we call the continuous capillaries. That's what we see here on the left in A. Basically, here's those endothelial cells right here. They reach out, they've got junctional complexes between each other, and they're sealed rather tightly to each other. Anything getting out of the blood or coming into the blood must cross the cytoplasm of that endothelial cell. That is a continuous capillary, and if I told you to design a capillary to serve this function, that's what you design, okay, is that continuous type of capillary. We don't want things to leak, and if we have an empty space there, we don't control as well what goes back and forth across there, do we? If it goes through a cell, we at least have some control over it, if it's passing across the cell membrane. And so that's continuous capillary. That is by far the most common type of capillary. The next type of capillary we talk about here we call fenestrated capillaries. And fenestrated capillaries have little tiny pores in them. And yeah, they are showing it here in the drawings, but it shows up much better when we look at the electron micrograph. Here is a continuous capillary, and a very small one. Wall's made up of one endothelial cell, that reaches up and folds up on itself, and the tips touch each other. And so here is this nucleus. If we look around, here is its cytoplasm going all the way around. They've got arrows pointing to these little tiny projections here. Do not worry about that. Just look, that cytoplasm is continuous all the way around, and you can see some organelle in the cytoplasm as well. That cytoplasm continues all the way around, isn't it? Whereas when we look at the fenestrated capillaries on this side, here's the nucleus. If we come around, and I'll come this way, that cytoplasm is much thinner, and there's places where there is no cytoplasm. That's what they're showing with these internal arrows. Little places where basically all I've got is basement membrane right there, but there's no cytoplasm, no cell membrane that molecules would have to pass, okay? That's what we call fenestrated capillaries, and those fenestrated capillaries basically have little pores in them. Things can leak out. What things will leak out, what the criteria determines if something leaks out or not, it's going to be molecular size. Most proteins are going to be too big to fit out through that. On the other hand, ions, amino acids, sugar molecules, okay, glucose molecules, let me say, all those will be able to leak out. Now, one thing students think about, that misconception students have, is all my glucose going to leak out or all my amino acids going to leak out. Think of this as a hose. If I've got a water hose here and I put fenestrations, little tiny pores in it, all my water doesn't leak out of those little tiny pores, does it? Some of the water squirts out of it, but most of the water is still going through the hose, correct? That's the same type of thing here. It's not like all my plasma is leaving, all my nutrients are leaving, and so on. It's that little tiny pinprick there that some of that water is leaking out as it goes past. Okay? Now, the other type that we have here, what we call sinusoidal capillaries. If I could be the editor of this, and they ask for my input on terms of this drawing. Sometimes when they ask me to review a book, they'll make different changes to drawings. Other times I won't when I suggest. I'd make the diameter larger. The sinusoids are going to be larger diameter. And when they're larger diameter, if you have normal capillary going into a sinusoid, what happens to a river when it goes narrow to be real wide? What happens to the current? It slows down, doesn't it? And so it's going to slow down. And then if you look at the wall of it here, think of these, think of this as being a puzzle where you take each of these puzzle pieces of endothelial cells and put them in place. If you think of a sinusoidal capillary, we took some of those little cells out, some of those little pieces out. They've got large chunks of missing in the wall, okay? Blood flow slows down at through sinusoids, but you really get exchange now between blood and the surrounding tissue, don't you? Okay? We're going to find these in a lot of places where we find these, we're going to have whole cells entering and leaving. For instance, if we look right here, all right, this is bone marrow. I think if you looked up here with the H's, you might recognize that as bone marrow. I've got adipose in here, but here's a sinusoid running right here. If you look at it, you pretty much get the idea, like, where's the wall of it? Okay, there's whole chunks of the wall missing. Why would you want a sinusoid there? Well, as those red blood cell and white blood cell precursors up there in the marrow become mature, they have a pretty easy time now getting into the bloodstream, don't they? Okay, and so you'll find it where you have whole cells. You'll find some endocrine glands. Most endocrine glands will have fenestrated capillaries, as will the kidneys and other places like that. But you'll find some endocrine glands, they want to be extra leaky. They've got sinusoidal capillaries in them. We'll see these in the liver, bone marrow, spleen, places where we'll talk about sinusoidal capillaries, and in many cases they're blood cells, whole cells entering and leaving circulation. Okay, now capillaries, you know, are going to drain into venules, and this is a slide that is nice because it goes from the smallest venules to the larger venules, all within one image right here. The venules, smallest ones are the post-capillary venules. Those are up at the top, labeled PCV. All right, and these post-capillary venules, if we take a look at them, they've got an endothelial cell lining, just like we talked about before, tunica intima, but then there's typically one layer of what looks like smooth muscle. And prior to 10 years ago, we'd always say it's got one layer of smooth muscle, tunica media. what they found out once they had antibodies for it and actually looked for it, those aren't smooth muscle fibers, those are pericytes. Okay? Remember, pericytes we now know can serve a contractile role. You find them just scattered here and there among the walls of capillaries. Once you get into the venule side, the postcapillary venules, that's where you're going to find the entire tunica media. It's not composed of smooth muscle fibers, it's composed of 1 or 2 layers of pericytes. Okay? Now these postcapillary venules are important here because the endothelial cells don't have the junctional complexes to hold them too tightly together, and these postcapillary venules are the primary place where your white blood cells will leave circulation. We talked about all exchange between tissue and blood occurs at the level of capillaries. Well, in terms of cellular exchange, it occurs more commonly at the postcapillary venules because their walls are designed for that. This is a slide right here showing where we've got a site of infection. This is the postcapillary venules, and these are tons of white blood cells that are squeezing out in the walls here of that postcapillary venule. Okay? I don't expect you to look at that and say, oh, it's a postcapillary venule. I tell you it is, and these are all white blood cells. Why are they all concentrated on the edge? Because that's where they're squeezing out between the walls of the cells of the wall and the postcapillary venules, not in capillaries. Okay? Now, postcapillary venules will drain into collecting venules, which are labeled CV. We do have some capillaries labeled C. I should have pointed those out up here earlier. If you take a look, multiple postcapillary venules have merged together into a collecting venule. Okay, and then the collecting venules will merge into a muscular venule. It turns out, we already said this has a tunica media of pericytes. The collecting venule has a tunica media of pericytes as well. And once we get to the muscular venule, that's where the wall is now composed of smooth muscle, like we think it should be, okay, like the rest of the blood vessels. Now, I don't expect you to be able, I'm not going to put an image up and say, identify the type of venule here. In all reality, the only way we could look at this is say postcapillary venule, collecting venule, muscular venule is the size of these, where you see them all right next to each other. If I just had one there by itself, where you can't compare them, you have no way of doing that. And so I'm simply going to expect you to label any of these as a venule, okay? However, I can ask you a question in terms of the order and why, what's the difference in the wall between a collecting venule and a muscular venule. One's got pericytes, one has smooth muscle. And so, yeah, you have to know what's in the notes here in terms of the order in which they go, the differences between them. You don't have to recognize the differences between them. The only way you're going to do that is if you had an antibody, say is it pericyte or smooth muscle, and when you compare sizes up here like you can in this image. All right, now when we get into veins, here is a vein right here. If we look at the one running, the one on the right running across, if you look at it, it's got a rather large lumen. You can see all the blood in it. You can see the nuclei would be the white blood cells. But for the size of the lumen, veins are going to have rather thin tunica media to them. They're going to have thin walls. Once again, you know that veins are low pressure. You lose virtually all your blood pressure when you go across the capillary bed. There's a number of capillary beds within the body. And then you know that you're going to rely on compression of skeletal muscle, compression of your respiratory mechanism, and valves. And here, right here, a little bit of a valve within the vein in order to return the blood back to the heart. Okay, if we're having to go against gravity. All right, we've talked about that in anatomy. The fact that a vein is such a low pressure system means that it's got a very thin wall. And a lot of times arteries and veins run next to each other. It's very obvious which one's the artery. It's got a thick wall. Very obvious which one is the vein. It's got a thin wall to it. And the vein typically has a larger lumen to it than that artery is going to have. And we're going to see that time and time again when we get into the lab. Here is a slide showing a valve a little bit better. You take a look, plenty of lumen, boy, they didn't really stain to show the wall of it, but you get the idea. It's a rather thin wall structure. Here is a valve we're seeing all the way, the flaps all the way to the end. Blood is gonna flow fine when it's going this direction. If it wants to go back the other direction, these two flaps are gonna close against each other and prevent backflow of the blood. All right, the last thing to talk about here then before the break will be the lymphatic system. I used this slide when we talked about the lymphatic system last semester, that it is a blind system of vessels beginning parallels the venous side of circulation, starting in the capillary beds, and it simply collects the fluid that leaks out of the plasma that leaks out of the capillaries. If you remember from physiology, you've got more that leaks out on this end and is collected back on this end, more that leaks out of the arterial side of capillaries than returns in the venous side of capillaries, and that extra interstitial fluid is going to be collected by the lymphatic system, going to be immunologically screened here within lymph nodes, eventually then passed back into circulation. And as it picks up that extra interstitial fluid, it will pick up pathogens potentially. That's why it's got to be immunologically screened. It will pick up debris. It will also pick up cancer cells if they have broken loose within that interstitial fluid. And so, this basically describes the function of it there. Lymphatic vessels are not pumped by the heart. Once again, they use that skeletal muscle compression as well as your respiratory mechanism in terms of pushing on the walls there, and they've got valves just like the veins have. If we take a look here, here's the idea of the capillary beds, the arterial side, the venous side of the capillary beds. You've got these blind-ended lymphatic capillaries here, which I won't go into the structure of the wall, but basically they have a portion here which isn't well attached, so that when there's greater pressure outside here in the interstitial fluid than there is within the lymphatic vessel, fluid's gonna leak in. That's how it first gets in, and then the skeletal muscle will move it along in terms of with the valves in order to move it back to circulation. This is looking at lymphatic vessels right here. We said veins were under low pressure, so they had thinner walls. Lymphatic vessels are even lower pressure, so they have even thinner walls. Here is a vein right here, and I know it's not a lymphatic vessel because I see blood in it. Okay. Occasionally, when the microtome comes across, it may, that's coming across here to cut the tissue to make the section. It may carry a red blood cell over here. You may see a red blood cell in a lymphatic vessel occasionally, but not like this. Okay, not a bunch of them. I know that's a vein, good-sized lumen. Look at the wall, virtually not present. I mean, very little structure to that wall, correct? This is a lymphatic vessel up here, no blood in it, but man, it looks just basically like an endothelial lining, doesn't it? I don't see any tunica media, anything within the wall of it there. That's kind of typical lymphatic vessel. Here's a lymphatic vessel right here, and here's a valve within that. Once again, lymph can move in this direction. If it wants to move in the other direction, those flaps will close against each other. Look at the wall of it. I mean, it basically just looks like a line. That's the going to be the cytoplasm of an endothelial cell making up the wall of that. I don't really see much wall below that. And lymphatic vessels often collapse. Here is one right down here. See how it's basically collapsed on each other? Basically collapsed. Here I can see nice endothelial cell nucleus right here. There's one there as well that's not flattened anymore. Here's one right above the pointer here, but a lot of times we're going to recognize it because we look in connective tissue. It looks like maybe just kind of an open area in connective tissue. Maybe it's separated a little bit upon, making that slide. So it's an artificial empty space there. If we look, we'll make out some endothelial cells occasionally on that lining. It's going to be a collapsed lymphatic vessel. And once again, we will see that in the lab here in just right after our break. Any questions at all on cardiovascular? All right, if not, let's go ahead and take a 10-minute break. And when we get back, we'll look at some